main objective of this video is to provide you with the theoretical basis of plant tissue culture, leading to demonstration of practical aspects where applicable. The whole video will take about 60 minutes, but is divided into several segments, so you can watch the segments as you progress through your tissue culture activities. To start with, let us look at the definition of plant tissue culture, or as is also known, the in vitro culture. It is the sterile culture of plant cells, tissues, or organs under conditions which lead to cell multiplication or regeneration of organs or whole plants. The important points to pick from this slide is that all microorganisms, such as fungi, bacteria, and viruses, as well as other pests, insects, nematodes, are excluded. That optimum environmental conditions with regard to physical as well as nutritional and hormonal factors is provided, and that the whole process occurs on a micro scale and in vitro. Of course, in vitro, which literally means in glass, came into being because initially, at least, glass vessels were used for the culture. There are several benefits attached to plant tissue culture, and these are summarized in the next slide. To start with, the number of genetically identical plants recovered from a single stock plant is greatly increased. Virus-indexed plants may be obtained through tissue culture, for example by meristem culture. Stock plants may be maintained in vitro, where glasshouse space for maintenance of plants is at a premium. This is referred to as germplasm maintenance. Tissue culture techniques may be used to obtain hybrids from incompatible species through embryo or ovule culture. Haploid plants may be obtained through anther culture. We can produce plants all year round using tissue culture techniques. Vegetative propagation of difficult to propagate species may be achieved by tissue culture. Production of new variants may be also achieved using mutagenic compounds or radiation. And finally, it is an important tool of research in many areas of plant sciences. The idea of plant tissue culture goes back to the turn of the century, when a German scientist put forward the theory of cell totipotency. Totipotency is the potential or inherent capacity of a plant cell or tissue to develop into an entire plant if suitably stimulated. Totipotency implies that all the information necessary for growth and reproduction of the organism is contained in the cell. Although theoretically all plant cells are totipotent, the meristematic cells are best able to express it. This means that every part of the plant has the ability to produce a complete plant and thus be used in the process of tissue culture. The next two slides demonstrate the parts of the plant that we can use as starting plant material, and as we can see from these slides, basically any part of the plant, from roots to stem to leaf, buds, floral parts, seeds and embryos, can be utilized as starting material. There are many types of in vitro culture. These are briefly summarized in the next slide. Culture of intact plants, for example, seed culture in orchids. Embryo culture, for example, immature embryo culture. Organ culture, for example, meristem culture, shoot tip culture, root culture, anther culture. Callus culture, single cell culture, and finally, protoplast culture. The area of micropropagation is, however, what we endeavor to cover in this video. Micropropagation is the true-to-type propagation of a selected genotype using in vitro techniques. Several stages are involved in micropropagation, each of which are influenced by an array of physical, nutritional, and hormonal factors. Micropropagation only makes sense when adequate starting material is used. Therefore, the choice of stock plant material cannot be done indiscriminately. For most ornamental and for fruit production, the starting material is an elite plant selected for some phenotypic characteristics. For example, the flavor of the fruit, the color of the flower, the shape of the bush, etc. In forestry and in vegetable production, the elite material will either be a selected phenotype, that means a specific plant, or this plant will be the bearer of elite seed, which will be used as starting material for a more or less extensive micropropagation program. The stages of micropropagation is summarized in the next slide. Stage 0, the preparatory stage. Stage 1, the initiation stage. Stage 2, the multiplication stage. Stage 3, the rooting stage, and finally, stage 4, the acclimatization stage. And we endeavor to take you through these stages by use of demonstration. Stage 0, or the preparatory stage, is about preparing the stock plant for micropropagation. Two principal considerations are involved in handling stock plants. One is reducing the potential for contaminant by fungi, bacteria, viruses, and other pathogens. The other is control of the physiological conditions of the starting plant material. This starting plant material is also called an explant. In general, stock plants should be healthy and actively growing, not in or entering dormancy. Pruning or use of supplementary light for increasing day length is sometimes useful in maintaining active growth. Ideally, stock plants should be grown in a glasshouse. Field-grown materials are often very difficult to disinfest and establish clean cultures in vitro. Not only growing plants in a glasshouse will reduce the population of microorganisms which are living on the surface of the plant, it also helps to produce quality plants for stage 1 due to regular fertilization, use of fungicide and insecticide. So, at the preparatory stage, we need to ensure that the stock plant is growing vigorously and is free from pests and diseases, especially the microscopic pests such as mites and thrips. By establishing the stock plants in a glasshouse, we can ensure that the plants have been fertilized properly, have been sprayed with pesticides and fungicides, and have been pruned so that new, young, vigorous growing shoots are established. Another important point is that under glasshouse conditions, we can control the humidity in the glasshouse. Generally speaking, a dry glasshouse will harbor less microorganisms. The hygiene in the glasshouse can also help to keep plants clean. As a general rule, plants destined for micropropagation should not be watered overhead. All the points we have just discussed will aid in reducing the infestation at the next stage, which is the initiation stage. So, to summarize to here, the objective of this stage of micropropagation is to produce healthy, vigorously growing plants free from pests and diseases. Now, let us look at stage 1, which is the initiation stage. The objective of this stage is to successfully introduce the desired part of the plant into the appropriate medium in the test tube or any other culture vessel that we use and produce clean cultures. A culture medium consists of all the mineral and organic nutrients that the plant requires for sustained growth. These are all the macro and micro elements, the vitamins and other growth factors such as myoinositol, glycine, nicotinic acid, pyridoxine, thiamine, etc. To this mixture, we also add sugar as source of energy and plant hormones when necessary, as well as a gelling agent. To prepare a batch of medium, we can either use pre-mixed media such as Morishige and Scooch, Gambur, White, etc., which can be purchased from chemical companies such as Sigma, which has a wide range of media in powdered form. The only thing we need to add to this mixture will be sugar, plant hormones, and the gelling agents where necessary. If, however, we are doing a lot of media preparation with various combinations and permutations of minerals and organics, we may need to opt for preparing our own stock solutions and use them as required. Whilst this provides a lot of flexibility, it may work out to be a bit impractical for small labs. One needs to have all the chemicals and a sensitive balance to measure accurately quantities required, a fridge to store the stock solutions, and suitable containers to store these stock solutions in. The final choice will very much depend on the quantities of the media you are preparing and the facilities in which you are working. To prepare media, simply measure the quantity of stock solutions or add the correct amount of powdered medium to deionized water. It is important that the water used for tissue culture be deionized. Deionized water can be purchased from many supermarkets or water purification companies. Large laboratories normally have their own purification plant. To this mixture, sugar is added and the pH is checked. As a general rule, the pH of tissue culture media is adjusted using either a weak acid solution like 1 normal hydrochloric acid or a base like 1 normal sodium hydroxide. A pH meter is necessary in big laboratories, but if you are a hobbyist and have no access to a pH meter, pH paper with narrow range would be just fine. As a general rule, the pH of most tissue culture media is set at around 5.6 to 5.9. The gelling agent is then added. There are various gelling agents that can be used. For example, agar, which is extracted from the cell walls of a number of general red algae, which produce an opaque gel and, generally speaking, is dissolved on a hot plate with magnetic stirrer for continuous stirring of the agar. Gelride is from bacterium Pseudomonas elodea, and gel cream from the walls of a cultivated red algae. Gelride and gel cream produce a clear gel and can easily be cold dispensed as long as the mixture is kept in suspension by constant stirring. Otherwise, the gelling agent will settle to the bottom of the beaker, resulting in a batch of medium which is not homogeneous. This means that some containers will have a lot of gelling agents and some will have very little in them. Once the gelling agent is added and dispersed or dissolved thoroughly, the medium can then be dispensed into culture vessels. There are a wide range of culture vessels: the glass jars, the plastic ones, which are autoclavable, the plastic ones, which are not autoclavable, and can be purchased in sterile packs. These have been sterilized using radiation. For these ones, media is bulk autoclaved, and then the correct quantity is dispensed into them under sterile conditions. For others, the culture vessels are capped and autoclaved for 20 minutes at 121 degrees centigrade at a pressure of 103 kilopascal. This pressure and heat will destroy all microorganisms which are in the medium and the culture vessels. household pressure cooker may be used instead of an autoclave in small hobbyist operations. Another method of sterilization of media is filter sterilization. In this case, a membrane, which is made of cellulose acetate or cellulose nitrate, with 0.22 micron pore is used. This is used only when we are dealing with compounds which are heat labile, such as some of the plant hormones used in tissue culture, for example, zeatin, gibberellic acid. All the instruments and equipment needing to be used must be either autoclaved or soaked in 95% alcohol. For example, the hypodermic syringe, the container in which the compound in question is going to be filtered into. The compound in question is drawn into a hypodermic syringe and then are passed through the filter like this in a laminar flow cabinet or any other clean environment.